Okay, so this particular lecture is going to go through Chapter 1 and be a lecture, and the PowerPoint that's associated with it. This PowerPoint is posted on the Blackboard course shell. It's labeled as Chapter 1. You can probably see a little bit of it from the recording, but you're welcome to print it out or to also have it open and kind of follow along as you're looking at the video. And what this chapter does is it kind of gives an overview of the entire semester. Gives you kind of a definition of biology, because it's a biology class that you're taking. Then kind of takes us through some of the organization of the semester and an overview, very general overview of some of the topics that we're going to talk about. So biology usually refers to study of life. Bio means living. Ology is the study of something, so this is study of living organisms. This chapter is going to give us a good overview of the major topics within the field of biology. Then we're going to go into each in a little bit more detail. So one of the things that your book goes through is the characteristics of life. How do you know that something is alive? How do you know that something is living? And so it takes you through some of these characteristics. Take in energy and materials from the environment. Sense and respond to the environment, grow, reproduce. Most living organisms maintain some kind of homeostasis, which is kind of a constant internal environment, and that can help them regulate and interact with the external environment. And then living things undergo change. When you're looking at living systems, things don't stay the same. Things are going to change over time. Now, one of the things that I really wanted to kind of emphasize is this idea that living things are organized. Living things have these different levels of organization. All living things begin at their most basic level being organized of atoms, at the atomic level of organization. All living things are made up of atoms. Even non-living things are made up of atoms, but all living things are made up of atoms. So to understand how living things do those things like grow, reproduce, respond to the environment, change over time, we have to understand what they're comprised of. We have to understand atoms and molecules that they're made of. Atoms can be put together to make molecules. Molecules form the structure of cells and are made by cells. Cells are the basic unit of life. Bacteria are single-celled organisms. Cells can be put together to make different kinds of tissues. So in human anatomy and physiology, we learn a lot about muscle tissue and nervous tissue. And those types of tissues can be organized into organs and organ systems. Cardiac muscle tissue can be organized into the heart, which runs the cardiovascular system. These are kind of the levels of organization of organisms, like a human being. So when we teach human anatomy, these are the levels of organizations that we teach. But in biology, we can stop not just with the organism, but with lots of other organisms. And so we can get into communities and populations and ecosystems and eventually to the entire biosphere. And that's kind of the roadmap for our semester. So all living things are made of atoms. We can put atoms together to make molecules. Those molecules are made by organelles in the cell, and those cells can be organized into tissues. They can then be organized into the actual organisms. But if we have a group of the same organisms, that's a population. If we have a group of living organisms, some are the same species, some might be different, that becomes a community. And then when we take into account everything, not just the living things, but the water resources, the air, physical resources in that area. That's an ecosystem. And when we look at a whole, across our whole planet, we can look at the biosphere, all of the ecosystems, all of the communities and populations of organisms within them, and these things that make up each of those organisms. So as we go through our semester, we're going to start by talking about atoms. Atoms and molecules and cells, because these are kind of the basic building blocks of all the organisms that are in our biosphere. Once we've talked about atoms and molecules and cells and what they do, that's going to take most of the first half of the semester. Then we can start looking at populations of organisms and how those populations change over time, and how different populations come together to make communities, and how communities live within different ecosystems, till by the last week, which on your syllabus we're looking at putting it all together, we can come back and kind of see how our semester has taught us these different levels of organization. Now, although this is a biology class, we're going to talk about some things that are not living things. We're going to talk about the non-living environment, because organisms continuously interact with the non-living environment. The interactions between organisms and their environment take place within an ecosystem. And when we talk about ecosystems, we can talk about nutrients and energy. All organisms need nutrients and energy. Your book shows you this figure to look at the cycling of energy and nutrients in an ecosystem. Usually, nutrients go through a cycle. So, for example, we're going to talk about a water cycle. The water cycle is things like rain comes down, condensation, it collects, it then evaporates into the clouds, it rains down, it collects, it evaporates, it rains down, it collects, it evaporates. That's a cycle. So that's represented in figure 1.3 as a cycle, a circle. Energy tends to move through systems. Energy comes into a system and gets used and can be changed in its form. Most ecosystems get their energy from the sun. We live on a planet in the solar system where the sun provides us lots and lots and lots of energy. Now, this isn't really a physics class, so we're not going to spend too much time on the different forms of energy and how energy can transform into those different processes. But we're all familiar with the fact that we get energy from the sun. Light energy and heat energy come from the sun. So inflow of energy into the system comes from light. It gets transformed in an ecosystem into chemical energy, and then it can leave the system as evaporated heat energy. There's another figure that I have in your PowerPoint. It's from a different textbook, but it gives a very similar view. So it has sunlight energy coming into this system. What makes this figure a little different, and the reason why I included it, is it includes some actual vocabulary that we'll use over the course of the semester: producers and consumers. So when we're hungry, we don't go sit in the sunshine. Humans can't make their own food energy from light energy. If we're hungry, we have to eat an animal, or we have to eat a plant, or we have to eat an animal that ate a plant, because plants have the capability of turning sunlight into food energy. called photosynthesis, and that's what plants are really, really good at doing. They take light energy and they turn it into glucose. So then we eat a plant, or we eat an animal that ate a plant. Humans are consumers. Animals, bacteria, protists, funguses, the types of organisms that can't go sit in the sunshine when they're hungry, become consumers of the organisms that can take light energy and produce it into food energy or nutrient energy. This still shows the one-way flow of energy and the cycling of nutrients from producers to consumers and back. So it still has a very similar information structure as the previous figure. But again, it produces this, or it includes this vocabulary that we're going to use of producers and consumers as we go through the semester. Once we talk about organisms and how they're organized as atoms and molecules, we'll then talk about their cells. Here's where we really get into life. Here's where we get into the things that are going to be unique about biology and not chemistry or physical sciences. We do have to learn some of that chemistry because it is the basic levels of organization of life. But here's where this biology class is going to diverge from some of the other classes that we teach. All cells contain DNA, so we're going to talk about DNA. And cells can either be prokaryotes or eukaryotes. And we're going to talk about what the differences between those things might be, what that might mean for different organisms. Here's a figure that we're going to use as we talk about cells that shows us some of the difference between prokaryotes and eukaryotes. And when we get to the chapter on cells, we're going to learn a lot about subcellular structures. That's one of the things that really sets apart eukaryotes from prokaryotes. These membrane-bound organelles that are inside of cells. There is a section in your book that we're not going to have a lot of time to go through this semester that talks about plants in the plant kingdom, fungi in the fungi kingdom, animals in the animal kingdom. What we will do is we'll kind of get an idea in this chapter about the fact that there are 3 major domains that we can organize living things into. From those domains, there are different kingdoms, and there's quite a bit of variety in different living organisms. If we look at this figure from your book, physically, structurally, and functionally, funguses are quite different from people. People are quite different from bacteria. And actually, funguses are quite different from bacteria. So there's quite a diversity in the kinds of living organisms that we're talking about as we go through the semester. So once we start out talking about atoms and molecules, then we move on to start talking about actual organisms, various different organisms. Then we can start to talk about a population of organisms of the same type. And what happens to that population as that population interacts with their environment. We will get into the idea that populations don't stay the same. Populations change over time. We'll get into the concept of natural selection. This is one of the basic tenets of biology. The fact that this is one mechanism for that change in populations. And it's based on a couple of principles that we have found to be true in almost all populations that we've studied. Members of a population have to compete for resources. Most of the time, populations don't exist in places where resources are completely unlimited. most of the time, there's some subtle competition for those limited resources. We'll come to this idea in the semester after we've done some things with genetics. So we'll have a basis for the tenet that members of a population inherit different traits. There's a genetic basis for natural selection. And so these two tenets lead to the fact that there's unequal reproductive success. Some traits are adaptive. They allow an organism to live in their environment a little bit better, to interact with that environment and regulate in a better way. Other individuals with different traits might not have that same reproductive success. The individuals that inherit traits that make them better suited to live in the environment are the ones that will survive and reproduce and leave the greatest number of offspring, the greatest number of genes. The very traits that enhance survival and reproductive success are going to be disproportionately represented in the next generation. So after we've studied genetics, here we can study something like allele frequencies in a population. And so that's where you're going to see that. I apologize that the text in this slide seems to be cut off. I might have to edit that on Blackboard so that you can see the rest of that phrase. So this is a figure that your book shows in this chapter, and we're going to come back and look at this figure as well as a couple other when we learn about natural selection. This figure starts with a population, group of the same kind of organisms. And this population has varied inherited traits. Natural selection has a genetic basis, an inherited basis. So these are bugs. Some of them are darkly colored and some of them are brightly colored. So that a bird doesn't have such keen eyesight. Birds don't have the same kind of eyes that we do. A bird comes along and a bird can see the lighter colored bugs better than the darker colored bugs. A bird can eat the lighter colored bugs better than the darker colored bugs. It's hard to eat something you can't see. So there is elimination of individuals with certain traits. Lighter colored bugs get eaten. It's hard to reproduce when you've been eaten. Only the ones that survive are the ones that reproduce. You have an increased frequency of the survival traits in the next generation. So this kind of gives an idea of what we're going to talk about as we talk about natural selection. The traits that enhance fitness get passed on to the next generation. Now, we play with this a little bit in artificial selection. Dog breeding is an example of artificial selection. So we take dogs that have certain traits and we breed those dogs, differential reproductive success, over time, selecting for the traits that we most want to see. And so that's how from wolves, dogs have descended to have these varied physical characteristics. Different breeds have a standard through different kennel clubs, and that standard very specifically describes the physical traits that a dog in that breed should have. We will do some different discussion boards and assignments that will relate to this idea of artificial selection. And then the last topic that this chapter goes into is something that's not really a preview of the rest of the semester, like the first part of the chapter. Here your chapter starts to talk about how we study science. How does biology or other types of science get studied? So this is where your chapter really starts to talk about the scientific method. The scientific method usually starts because someone makes some kind of observation. They notice something. Leaves on trees turn green. That can often lead to a question. Why are the leaves on the trees green? One key part is to develop a hypothesis. I think that the leaves on trees are green because there's a green molecule inside them. So you devise an experiment to test the hypothesis. I'm going to take leaves, I'm going to chunk them up into little small pieces, and I'm going to try to figure out a way to separate the molecules in them and see if there's a green one. I'm going to make a prediction. I'm going to see a green one when I do that. So then you do the test. You go find some green leaves, you put them in the blender, you break them up until they're really, really small pieces. It looks almost liquidy because you can't see the individual molecules. Then you take some filter paper, like a coffee filter, cut it into a strip, and you put some of those molecules on there, and you put one end of the filter paper in water. You let the water go through and separate out the chemicals. It's called liquid chromatography, but it's something that you could very easily do in your kitchen. Maybe you try out different kinds of leaves. Maybe you do some broccoli leaves and some lettuce leaves, some cabbage leaves. Maybe you do some other types of cabbage, purple cabbage, red cabbage, yellow cabbage, green cabbage. And you start to see when you separate the molecules, when the water runs across the filter paper and separates the molecules, that not only are there green molecules, but there's yellow ones, or red ones, or purple ones. And so that gives you the results, tells you whether your hypothesis was correct, and might even give you some ideas for some future questions and problems to explore. Because your observations in your experiment can start the next round of the scientific method. So they give an example. Observation, my flashlight won't work. I wonder what's wrong with my flashlight. Let me try a few things. Maybe the batteries are dead. It's a hypothesis. If I replace the batteries, they'll start to work. So I replace the batteries. Turn on the flashlight and it works. The results supported the hypothesis. Or it didn't work. Okay, maybe it's not the batteries. Maybe it's the light bulb. That's a revision of the hypothesis. Scientific method can sound very different from things that we do in our normal lives. But scientific method is really exactly what we do in our normal lives. When we have a problem or a question, and we try something out to see if it answers the question. When remotes are dead, when flashlights are dead, when car batteries don't work, we're constantly doing the scientific method. We're constantly making a hypothesis and testing the hypothesis to see if what we think is really the case. So I don't want you to get too intimidated by scientific method. Scientific method is a process that we all go through. It just has some additional vocabulary to it that we're not as used to using. So whatever it is that we're testing is the variable. The things that can change or can vary is the variable. If in the flashlight experiment you changed both the battery and the lamp at the same time, and the flashlight worked, you wouldn't know which of those was the problem in the beginning. So first you changed the batteries, and then you changed the light bulb. What we do is we go through the process step by step, only testing one variable at a time. Experiments are designed to simplify these complex systems by only looking at one variable at a time. And the experiment has to be designed to either support or falsify your prediction. So if your prediction is, I think the batteries are dead, you don't change the light bulb. You change the batteries. That's the test that supports or falsifies your prediction about the batteries. A lot of times, we may want to have a control. We may want to control for our variable, especially in biological systems when there can be lots of things going on. We need to try to have some sort of a control that tells us what's going on with our experimental group. When we do drug research, a lot of times drugs have to be packaged in some sort of way that we can have a person take the drug. So for example, the drug is in the form of cream. There's some sort of skin disorder. A doctor develops a drug that he thinks will help it. They put the drug in a cream, put the cream on the person, and their skin is all better. Well, how do you know it was the drug and not the cream? It's like the flashlight. We don't change the batteries and the lamp bulb at the same time. So how do we piece out just what the effect of the drug might be? We can have another group that gets just the cream that the drug is in. Without any drug, just the cream base. Now we have two groups of people with this skin disorder. Half get just the cream, and half get the cream with the drug. If the improvement is because of the cream, both groups will get better. But if the improvement is just the drug, then just the drug group gets better. So that experiment is designed to specifically support the hypothesis that that one variable, that the drug, is what is making the effect. Now, I really want to address, as we're talking about scientific method, the use of the term theory. A lot of people think that the term theory in biology means that something is not proven. And that's actually the opposite of the truth. A theory is something that has so much scientific evidence and so much scientific support that we elevate it from the level of hypothesis to the level of theory that has all of this evidence to support it. The only reason that we use the term theory is the fact that scientists know that there are lots of things that are yet to be discovered, and we don't assume to know what they are. In the 1930s, we didn't even know the structure of DNA. That didn't come along until the 1950s. So in the 1930s, we had theories. Some were confirmed, and some were changed by the future evidence that we had in the 50s. At this point, all of the evidence that we have ever gotten show us that organisms are made of one or more cells. But we're sending robots to other planets. There's a chance we'll find a living organism that's made of something other than cells. But right now the theory is all organisms are made of cells. All matter contains atoms. Change happens over time to populations. Microorganisms can infect us and cause disease. Human activities are causing Earth's temperature to increase. There's overwhelming scientific evidence for all of these theories. All of these theories have gone through this rigorous process of observations and questions and experiments designed to test them with controls. And every time that happens, we find the hypothesis supports the theory. So theory doesn't really mean guess. Theory doesn't mean the same thing as hypothesis. Theories are accepted scientific tenets that have the overwhelming support of the research evidence that we have to date. So this is a figure from a different textbook that shows some of those theories. Your book goes through some of that same information, that same definition in the chapter, as it talks about this idea of scientific method and how we arrive at theories, the difference between a hypothesis and a theory. The other thing that I want to end this lecture by going through that also relates to the scientific method is the idea of critical thinking. Critical thinking is being critical, being a critic, really thinking about and analyzing. So as you get data from your experiment, and as you get data to support your hypothesis, and as you get more and more and more hypotheses that are supported by data, that it turns into a theory, that's all the result of analysis and critical thinking. And so I've gotten a little bit into the PowerPoint about some ways that we look at critical thinking. This is a critical thinking checklist that is a figure from a different textbook that you can look at. And then on the last page, there's a link to a web page that goes through the development of levels of critical thinking. This is something called Bloom's taxonomy. Bloom's taxonomy is something that we talk about a lot in education because part of the role of education is to help facilitate critical thinking. But this is a really helpful way to think about evolving different levels, higher levels of critical thinking. So we start with remembering. Can you recall information? This is rote memorization. This is regurgitation. This is what children do when they first learn a word. They say the word exactly the way they've heard it. They don't even know what the word means. They can just repeat it. Then the next level is understanding. Do you understand what it is that you've memorized? Children do this when they start to understand how to use a word, and they're not just saying the same sounds that they've heard. The next level is applying. Can you apply the information? Can you use that information in a new way? Here's where you start to be able to answer test questions. Can you take the information that you've learned and answer a question? Can you take the information that you've learned and apply it to a discussion board prompt or an assignment? Analysis. Can the student distinguish between different parts? So when we get into conversations about subcellular organelles, do you understand the difference between functions of the different organelles? Do you understand the difference between the different functions of a cell? Here we're really starting to get into some of the higher levels of being able to use information, apply it, and analyze it. The next level is evaluating. Argue, defend, support, evaluate. So here's where we really start to take a principle that has some scientific basis and we evaluate that scientific basis for that thought. And then creating. Can the student create new information? In the sciences, this is usually research, graduate level work, where you actually get new data and have new information, and you have figured out a way to understand it and apply it and analyze it, evaluate if it's good data, and then put it forth to support the hypothesis. So this is helpful to think about how we can do things this semester in the class, or as you do the scientific method assignment. So as you learn any new concept, you're going to start over with remembering. You have to remember the terms. You have to remember the vocabulary. You have to remember the word and understanding what it means. Then once you remember the term, do you understand the concept behind the term? Can you use that concept? Can you apply that concept? And can you analyze a situation using that concept? So those are the kinds of things, the kind of goals to set this semester with each of the concepts that we're talking about. So with this concept about scientific method, can you remember the steps? Do you understand how those steps fit together into an overall process? And then can you use that process to do the scientific method assignment? So this gives us a really good paradigm to kind of look at that. Now, if we go back to the scientific method, talk just a little about the scientific method experiments that you're going to be doing. I don't want you to get too intimidated by the scientific method experiment. Your experiment doesn't have to be something novel that nobody's done before. It doesn't have to be something that's completely creative or outlandish or over the top or costs you $1,000 to do, because the purpose of the assignment is to take you through these steps and then to get you to write your thoughts out to help you understand the steps. So I'll give you an example of an experiment that probably takes more than a week to do, but you could look at, does different colored light affect plant growth? That's a question. You can start with an observation about your plant growing, and you can think to yourself about an experiment to do. The question, what kinds of things will affect plant growth? Does different color light affect plant growth? So you take your plant, you split it into two, put it in two different pots. You put a red light bulb near one and a regular light bulb near the other, and you measure their growth over time. And maybe you conclude that the red light bulb caused it to grow faster. What we're doing is taking you through the steps of, here's an observation about my plant growing. Here's a question I have. What might make my plant grow faster? Does the light that my plant gets change the rate at which it grows? The hypothesis, red light will make my plant grow faster. And then you do the experiment with the controls, and you see if your data supports your hypothesis. The hypothesis just picks one thing as the outcome. You could just as easily say, I think that the white light bulb will cause faster plant growth. If the data doesn't support that, that tells you the red light bulb caused more plant growth. So it's actually not a bad thing when the data doesn't support your hypothesis. It just tells you the other thing is true. There's a number of different things that you can look at doing. Anytime you're in the kitchen and you think, I wonder why that works. I wonder if that would work if I did it differently. Think of the questions that you've always wondered about. Then think if you can design an experiment to answer the question. That's what the assignment is really getting at. When you write your results, write them so that somebody else could just pick up your paper and do the exact same experiment that you did. When you write the introduction, give me that background. Give me those thoughts. Well, I was in the kitchen one day, and I was whipping cream, and I wondered if I added something else to it, would it change the way that I did the whipping cream? Or I was in the backyard, and I was wondering if I covered up a plant, if it would still grow. Give me that thought process that led to your question. Give me those observations. Take me through this whole process. What were the observations? What were the questions? What's your hypothesis? I think if I cover up that plant, it's not going to grow. Okay, so I covered up the plant, and it didn't grow. Tell me about your controls. Well, I had another plant that wasn't covered up, and it grew. The question to ask yourself to get to the controls is, How do I know that the effect I saw was truly because of the variable? With the drug and the cream, how do we know the effect was truly the drug and not just the cream? With the plants, why do we have two different colored light bulbs? Is it just a light bulb that's there, or is it the actual color of the light bulb? So anywhere you can, think about ways that you can have a control for your variable. There's no certain length. The guidelines really just say to put things under the headings. Some things will be longer than others. Some things will be shorter than others. Because it really depends on how you do the experiment, questions that you're asking, the hypothesis that you come up with. My goal is really just to motivate you to take yourself step by step, explicitly through the process so that you can start to understand the steps. So if you have questions, please be sure to put them on the discussion board or send me an email. And I will be making comments and sending things back to you so that you can kind of get some feedback on what you did when you submitted your assignment. As always, send me an email if you have any questions or post them on the discussion board. And I'll be getting next week's material up pretty soon, Chapter 2 and Chapter 3.